Welcome to the Emery Medical Genetics review series. In this series, we make the concepts from Emery Medical Genetics 16th edition, which is the latest edition, clear and connected. Think of it as a roadmap. Instead of reading chapter by chapter, we bring together discussions scattered across the book into one integrated concept, so you see the whole picture at once. These videos are designed for biology undergraduates preparing for advanced studies, medical students mastering their core reference, and graduates or professionals refreshing their knowledge. You can use this series in different ways. Watch first for an overview, learn alongside the textbook, or return later for quick review. Let's dive into today's concept. Hello everybody! Today, we want to recap essential protein-making steps, which are transcription and translation. Let's begin with this question. In the previous video, we learned about the structure of DNA and how it replicates. But how do we get from DNA to RNA and protein as the final product? As we know from the central dogma, genetic information flows from DNA to RNA to protein. These steps involve transcription and translation that we want to get to know them right now. Transcription. When the cell needs to build a specific protein, it doesn't take the entire blueprint out of the nucleus. Instead, it creates a temporary single-page work order. This process of making a working copy is called transcription, and the resulting molecule is a messenger RNA or mRNA for short. The transcription process involves a highly important key enzyme called RNA polymerase. This enzyme acts like a scribe, which means it can read one of the DNA strands, which we call it the template strand, and this enzyme, RNA polymerase, can build a complementary mRNA molecule from this template DNA strand. The newly synthesized mRNA is a copy of the other DNA strand with one key difference. RNA uses the base uracil instead of the thymine, which is found in the DNA. So, where there is an A in the DNA template, the RNA polymerase places a U. Actually, but the resulting mRNA molecule that is just created, it's not ready to go to the protein-making factories just yet. It's what we call it a raw transcript, and it needs to be processed before it can leave the nucleus. This is a crucial editing process that involves three main steps: splicing, 5' capping, and polyadenylation. Splicing. Genes are not continuous coding sequences. They contain non-coding regions called introns and coding regions called exons. In the splicing process, the introns are precisely cut out and the exons are joined together. This process, which is splicing, is a highly accurate process that ensures the final message makes sense. 5' capping. In this process, a special chemical cap is added to the beginning of the mRNA molecule. This cap serves three main important purposes for the final mRNA molecule. It can help the mRNA be transported out of the nucleus, and it helps the mRNA molecule to be able to attach to the ribosome for translation process. And finally, this cap can protect the mRNA molecule from being broken down by enzymes that can degrade the mRNA molecule. Polyadenylation. After the main part of the mRNA is transcribed, a long chain of adenine bases, which we call it a poly-A tail, is added to the end of the mRNA molecule. This tail provides stability and it can protect the mRNA from degradation once the mRNA molecule is out of the nucleus into the cytoplasm. So, we've learned that RNA is made from the part of DNA we need during transcription. Now, in the next step, from this RNA during the translation process, protein will be made with the help of translation machinery. This entire process takes place outside the nucleus in the cytoplasm at specialized cellular factories called ribosomes. For translation to occur, we need three key players: the mRNA molecule, ribosomes, which are the factory of translation, and the tRNA molecule. About the mRNA molecule, which is the work order, this is the message we just created through the transcription process. This molecule, which is mRNA, carries the code that dictates the exact sequence of amino acids needed for the protein. The ribosome, which is the factory of translation, these complex machines are composed of ribosomal RNA or rRNA for short and proteins. They are the site where the actual protein assembly takes place. The tRNA, these are small adapter molecules, and each tRNA is responsible for picking up a specific amino acid, which is the protein building block. And the tRNA molecule delivers its amino acid to the ribosome according to the mRNA's instruction. About the translation process, actually, translation works much like an assembly line, which means it can build a polypeptide chain one amino acid at a time. At the first step, the mRNA strand feeds through the ribosome. The ribosome reads the mRNA sequence in 3 base units called codons. And in the next step, as the ribosome reads the codon, the correct tRNA molecule, which carries its specific amino acid, can recognize the specific codon and temporarily binds to that codon. And in the final step, the amino acids carried by the tRNA molecule are then linked together by peptide bonds, which can form a growing chain of polypeptides. And so the ribosome moves along the mRNA. It can constantly add to this chain until it reaches a stop signal. But again, once the long peptide chain is released from the ribosome, it is rarely functional immediately. Just what we had in the transcription process, that we had made raw transcript of mRNA molecule, and we had to perform some editing important process. Here in the translation process, again, we have to edit the final protein to this protein be able to be functional in the cell. This process of post-translational modification is the final assembly and quality control step, which is essential for a protein to become a mature and active protein. But it is important to understand that we have some exceptions throughout the biology, and we have some proteins that are mature immediately after they are made from the ribosome, but however, it is rarely expected. These modifications in post-translational modification include folding, chemical alterations, cleavage, and localization. In the folding, the resulted chain of polypeptide bonds must spontaneously or with help fold into a complex and specific three-dimensional shape. This shape is absolutely critical for the final protein function. So, in the folding process, the protein is finding its final and functional shape. In the chemical alteration process, various chemical groups like phosphate or sugar molecules may be added to specific amino acids to change the protein's activity. In the cleavage process, sometimes a large inactive protein precursor is cut into smaller and active pieces. A classic example of this process is the cleavage of proinsulin into the active hormone, which is insulin. And finally, in the localization process, the mature protein is shipped to its correct destination within the cell, or it is prepared for secretion outside the cell. All in all, translation is the process of precisely decoding the mRNA message into a chain of amino acids, which is followed by the essential modifications that transform that chain into a functional worker of the cell, which is the final protein. But how does the ribosome know which amino acid to bring? That's because of the genetic code, the universal dictionary of life. This code is simply the set of rules your cells use to translate the 4-letter language of DNA or RNA into the 20-letter language of proteins. The fundamental unit of this language is called the codon. A codon is a sequence of 3 nucleotide bases. Each 3-letter codon specifies a single amino acid. Think about the math for a second. We only have 4 letters in our RNA alphabet, which are A, U, G, and C. If the code used just 1 or 2 letters, we wouldn't have enough combinations to name all 20 amino acids. But a triplet code gives us 4 by 4 by 4 or 64 possible combinations. That's why we have triplet codon role. However, that's way more than enough. 64 combinations possible is way far more than 20 amino acids that we have. This success leads to some of the code's most important features. First, redundancy or degeneracy. This is the most critical feature. Since we have 64 codons but only 20 amino acids, most amino acids are coded by more than one codon. This is essentially the code's safety net. If a mutation happens and changes a single base, the redundancy means the cell might still end up incorporating the correct amino acid into the protein. So this feature, which is redundancy or degeneracy, it's a built-in error check that makes the system incredibly resilient against improbable mutations. Second, non-overlapping. The ribosome is very strict about how it reads the message. It reads the codons sequentially, one right after the other, with absolutely no overlap. This establishes the reading frame, and it's critical because shifting this frame by just one base is like throwing a wrench into the whole sequence. It's completely changed the final product. Third, start and stop signals. Just like any good language, the genetic code has punctuation. The codon AUG acts as the universal start codon, which tells the ribosome exactly where the protein synthesis should begin. And conversely, specific codons act as stop codons, which they can signal the precise end of polypeptide chain. Amazingly, the genetic code is almost universal across nearly all life on Earth, from simple bacteria all the way up to humans. This shared code is one of the most powerful pieces of evidence that we all share a common evolutionary ancestor. But there are tiny exceptions. The most notable difference shows up when we compare the code in the cell's nucleus to the one in the mitochondria. The mitochondrial genetic code has slight tweaks, including different stop signals. It's also more streamlined. The mitochondrial DNA is more streamlined, having fewer types of the tRNA molecules. It relies on the famous wobble hypothesis, which refers to a bit of flexibility in how the third base of a codon is read to efficiently decode the entire sequence. So, in short, the genetic code is beautifully elegant and robust dictionary that guarantees the precise and efficient production of every protein in your body. Its built-in redundancy and crystal clear rules are what make the whole system so durable. Congratulations, you've made it! Every video you watch is a step forward in mastering genetics. Keep going. Your future expertise is already taking shape.